This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 50 for September 18th, 2009. Hey everybody, it's Vincent Racaniello, and I can't believe it. We've reached episode 50. Congratulations, TWiV, and Dick Despommier is not here. But I do have today someone equally as good and maybe even better. It's Jason Rodriguez. Welcome, Jason.
Hi, how's it going? Hi, Vince.
Jason's a postdoctoral scientist in the lab of Steve Goff, which is my neighbor here on the 13th floor at Columbia. Known you for a couple of years now.
Actually, I've known you much longer than that.
I was just at a meeting. I was at a thesis defense earlier, and one of the persons on the defense said they knew me for many years. I couldn't remember. So the problem was when you get old, you can't remember who. Once I was standing in line at an airport, and a guy came up to me and started a conversation for 10 minutes. I had no idea who he was. He said he knew me, and he went off, and I didn't know who it was in the end.
See, Vince, you're more famous than you think you are.
I don't think so. I just know about a dozen people, and the other dozen I forget. Anyway, Jason's a postdoc, as I said. He does research on, or has done some research on, a virus we're going to talk about today, XMRV virus. So we're going to get the scoop from Jason on that. Jason, tell us a little bit about your scientific training. Where did you get educated?
Well, I started off at the State University of New York, Geneseo, for biochemistry. And I quickly decided that New York City is the place to be for science and for other exciting things. And I did my PhD at Mount Sinai School of Medicine.
Is that right?
Yep.
That's where I did mine,
I know, with Peter Palese.
On the same floor, 16th floor?
No, I was actually in the East Building.
Okay.
So the new building over there.
Of course, you're a bit younger than I am. Significantly.
Well, that's how I know you, though, is because I would talk to Peter Palese, and he says, "Oh, you're going to go to Columbia?" Well, he's a good guy in the
Who was your mentor there at Mount Sinai?
Dr. Kurt Horvath.
Okay.
So he was a big interferon signaling guy. And I did my PhD on a few viruses, paramyxoviruses, that actually block interferon signaling.
And then after that, you came here.
Yes.
But did you go with Dr. Horvath to Northwestern?
I did. He decided to move his lab towards the tail end of my PhD, so I transplanted myself along with the lab to Chicago, which was fun.
For how long? How long were you there?
I was there for about 2 years. I defended my PhD in New York, and then went back for a little longer to finish up a few projects.
So there you worked on what viruses?
Worked on Nipah and Hendra virus. So along with XMRV, I tend to gravitate towards these emerging viruses.
And someday, I've always wanted to have you come and talk about Nipah and Hendra, but the XMRV story broke last week, so we'll do that first, and we'll get you back another time, because Nipah and Hendra is always very interesting, and we need to have someone who knows what they're talking about. So today we're going to talk about XMRV, as you've heard. But before we do, I want to talk just briefly, maybe 15 minutes or so, about a few influenza stories that I think everyone needs to hear. It's interesting information. And, of course, they're all about the 2009 pandemic H1N1 virus.
Which has got a lot of press, by the way.
Today?
Just in general.
In general, it is. Absolutely. I mean, have you ever seen anything like this in your career for any virus?
Other than HIV.
Yeah, that's true. In the beginning
days.
In
the beginning, and maybe XMRV now might be getting...
SARS too, probably.
Corona SARS, yes.
That was gone very quickly, though.
Yes.
But this will persist. Yeah, the press is going crazy with this. What I'm amazed is that so many papers being published very quickly.
Yes.
A week or two of review, and that's it. And a couple of the ones today were all rushed in. Everyone wants to publish first. But the first paper we have today is from New England Journal of Medicine. Response after one dose of a monovalent influenza A H1N1 2009 vaccine preliminary report. So this is a clinical trial where they inoculated volunteers with an inactivated influenza vaccine.
So can you explain what monovalent?
Monovalent means it just has one subtype of influenza virus, H1N1. There is actually now a trivalent vaccine available. It's 3 different strains. There's an H3N2, an old H1N1, and an influenza B. So we call that a trivalent. You know, there are tetra, deca, dodeca. There are some 25 valent bacterial vaccines with 25 different serotypes in them.
That's very complex.
You know, there are 100 different rhinoviruses, so I wonder if someday could actually make a 100 serotype vaccine. You know, at some point you have such small amounts of each strain that it's probably not immunogenic anymore. I don't know what point that comes. It depends on the antigen, I guess.
Well, a lot of people that suffer from the common cold would love to hear that news.
Someday, not anytime soon, though. It's going to be a while. But vaccinology is improving, so we'll eventually get there. And this monovalent, that's the 2009 pandemic vaccine. It's going to be monovalent because there wasn't enough time to test it in combination with anything else.
Right.
And this is the first of the tests in people of a vaccine. This was made in Australia. And this is a vaccine that's grown in eggs. It's inactivated with formalin and treated with detergent. And then it's injected into your arm. And here they used, I think, 240 volunteers. And the question is, do they make antibodies that would be protective after 1 or 2 doses? So the whole trial is actually a 2-dose regimen. They inject them, and then at 21 days, they took serum, and then they gave them another injection, and they're going to wait another couple of weeks.
And my understanding is that it's pretty effective. A lot of the subjects actually made antibodies.
Yeah, in this study of these 240 subjects, so they split them in half, less than 50 years old and greater than 50 years old. And then they split them further. They gave some of them 15 microgram dose and some of them 30 microgram. So the typical flu vaccine is 15 micrograms. They wanted to try 2 different levels. And yeah, like 97% made an immune response, which they think would be protective. And so we should go into that just a bit. So what they do is they immunize people with the vaccine. They wait 21 days, and then they take some serum. And this is the report after that first serum bleed. So there's going to be another report in a couple of weeks, presumably. And what they do is they do 2 kinds of tests. They do what's called a hemagglutination inhibition assay, an HI assay, and a microneutralization assay. These are 2 different tests. They measure 2 different things. So when you put influenza viruses into you, you make antibodies against them. And you know influenza viruses, of course, are enveloped. They have glycoproteins in the envelope. One of them is called the hemagglutinin. The other is the neuraminidase. But most of the important antibodies are probably directed against the hemagglutinin.
It's the one of the most antigenic components of influenza.
That's right. And that's the one that attaches to the cell receptor. Now, the cell receptor is sialic acid. And it turns out that red blood cells have a lot of sialic acid on their surface. So if you add influenza virus to red blood cells, it doesn't infect them. It sticks to them, and the red blood cells all stick together. So if you put these red blood cells in a tube or in a 96-well plate, it forms a nice lattice, which is called hemagglutination. If there's no influenza virus in the well and you just add red blood cells, they tumble down the side and they make a little button down at the bottom of the well. So you can actually roughly determine how much influenza virus is in a sample by making dilutions of the virus and mixing it with red blood cells, and you can see how far out of the dilution series you get hemagglutination.
Right.
So that's a hemagglutination or an HA assay. I did so many of those when I was a PhD student at Mount Sinai with Peter Palese. I could do them in my sleep. Now, a hemagglutination inhibition assay, you do the same thing except you use 1 dilution of virus, and you add serum, and you make dilutions of the serum. And at high dilutions of serum, it's going to block the virus attaching to the red blood cells because some of the antibodies against the hemagglutininin will block that interaction. And then you find that the point where you no longer get inhibition of hemagglutination, and that's the titer of the antibody. Generally, people think that a titer of 1 to 40, that means at a dilution of 1 to 40, you don't get any more hemagglutination inhibition. 1 to 40 correlates with protection.
Okay.
It's a rough number. So if you generate a 1 to 40 titer in this trial, for example, or better, It's likely that the antibodies are going to be protective, and it means that that dose of vaccine is probably good.
And that's correlated with like past studies where they show that that dilution actually does have a protective effect, correct?
Yes, it's correlated with past studies which showed effects. It's also based on a mathematical model.
Oh, okay.
Which they massage the numbers and come out with it, yeah. So in this study, titers of 1 to 40 or more were attained in 97% of the people that got 15 micrograms and 93% who got 30. So basically the same with both. So you don't need 30 micrograms. 15 is enough. The neat thing is there's no adjuvant here.
Exactly.
And I don't know if you've been listening, but people are all upset about adjuvants.
Right. Well, I know adjuvants from my immunology class.
Did you ever have one?
I don't know. I haven't.
Yeah, I've never had a vaccine with an adjuvant. And people are understandably afraid, especially if something they don't understand very well. And we don't understand adjuvants all that well. So the public has every right to be afraid.
So an adjuvant, if I remember correctly, a couple years ago, isn't it something that also activates the immune response along with the vaccine so that the antigen or whatever you're vaccinating against, it actually provides a bigger boost to the immune system, thus creating more antibodies?
Right. And do you know how some of these adjuvants are thought to work?
I don't.
They actually stimulate
interferon.
They are TLR. Some of them are TLR agonists, or they somehow trigger interferon, and that helps to mature the adaptive response. So it's really interesting.
It all goes back to interferon.
It does. It does. So it prepares the adaptive response, so you need less antigen. But in this trial, they say we don't need any adjuvant, and that's nice because people are afraid of it. And no matter what you say, you could cite any clinical trial, people will still be afraid, I think. So I think it would be best in the U.S. to not use adjuvant. Although, in Europe, adjuvants are routinely used, and there's really not a problem. But
it probably is associated with, I would assume, any adverse effects.
It's very difficult. If it's a rare event, you have to have a very large clinical trial. And this is 240 people. You're not going to pick up a rare event. 10,000, 20,000, 30,000 people you might need. But what if the event is 1.5 million? Very difficult to pick up. So,
speaking of adverse effects, this paper actually shows that there really wasn't many at all. Local discomfort in the injection site and some headaches, but it was pretty good.
Let's see what they said here. So yes, redness or soreness at the injection site. No deaths.
That's a good thing.
Always good news. This is very interesting. We should just mention this. Stopping rules were not triggered.
Now, what is that? What are stopping rules?
So in any clinical trial, you have to file a very detailed protocol before you start. And you have to say, what will stop this trial? What kind of adverse effect will stop it?
Like death.
Death would certainly be one of them. But a terrible anaphylactic reaction, that's a stopping rule. So they never met those here. They don't tell us what they were, but you can actually find all the rules for clinical trials online. So you can see what the stopping rules were. Let's see what kind of all right. Headache, malaise, myalgia, muscle.
Which is just a fancy word for just feeling bad.
That's right. 30%. Systemic adverse events, 30%. Nausea. 3 subjects had influenza-like illness. So the interesting thing here, aside from the soreness at the injection site, or maybe a little bit of fever, everything else is just stuff that happens normally. Right. And it just so happens that these people got a experimental vaccine.
Basically. Getting a vaccine is basically generating an immune response. And part of that immune response is feeling kind of crappy when you get sick.
And you get fever sometimes, right? And the injection site is often sore. You're putting a lot of antigen in. I read in another trial, someone had diarrhea. They probably would have had it whether or not they got the vaccine. These are associated events. They're not caused by the vaccine. If you wanted to know if they were caused, you'd have to do a much larger study.
Right. Their sample size definitely needs to be
bigger. You'd have to do a control group, which they didn't do here, which doesn't get any vaccine. So the purpose of this trial is to say, is this dose of vaccine enough to make antibodies that would protect? And it's not to really look rigorously at the side effects. To do that, you need a lot of unvaccinated people. So you get the baseline for diarrhea, headaches, and all that stuff, right, in the population. This is actually not my field. This is clinical trial is a science in itself, and there are people here at every medical center that do these for a living, as you probably know.
Yes.
It's totally, completely essential for any vaccine or drug, but...
do we see a lot of these type of studies here at Columbia? I would assume we would.
Dozens.
Yeah.
In fact, there are many HIV clinical trials run out of Columbia here. Scott Hammer does that. Very important stuff, looking for vaccines.
Right.
Various combinations of adenovirus vectors and DNA vaccines and so forth. And, you have they're hard to do because you have to get volunteers. And often the people have to come back several times. It's not easy. When I read these, I get scared because I would never want to do this. The number of people involved, besides the people who plan it, there are nurses, pharmacologists. I mean, it's just data massagers, administrators. It's huge. And these are incredibly expensive things. I just like doing my virus experiments.
But it does need to be done at some point. So
absolutely has to be done. And we're grateful to the people who do it. And this one is a good result. One dose without adjuvant, 15 micrograms. This is done in Australia, by the way. And the vaccine was made by a company called CMAX.
Never heard of them.
I'm sorry. That's the company who did the clinical trial. The vaccine was produced by CSL Biotherapies, Parkville, Australia. Okay. But Jason, you'll be interested. All the assays, hemagglutination inhibition, microneutralization, which we didn't explain. They also look for virus and nasal. They subcontracted all of that out.
Oh, wow.
So there are companies that just do this for clinical trials. Right?
That's quite a specificity.
That's the kind of thing you can do with influenza because it's such a big deal. Microneutralization, you take the same sera, and instead of looking at whether it inhibits virus binding to red blood cells, you ask if it inhibits infection in cells. You do basically a plaque assay with serum in the overlay.
And do you think that would be a more meaningful result as compared to the antibody?
That's a good question because, the HI antibodies are just measuring blocking of attachment.
Right.
But we know for influenza that neutralization is also achieved by antibodies that don't block attachment. They block uncoating.
Right.
So I think the microneutralization is probably more meaningful. But, over the years, they've had a lot of experience with HI, and it does correlate well with protection.
Okay.
But you notice they did both here.
Yeah.
And that's probably why, just to be careful. All right, so that's the one study I wanted to mention. Now, there is another ongoing study, and I find this very interesting. This is a trial of the same vaccine produced by another company with adjuvant.
And what was the adjuvant that they used? I don't quite recognize it.
MF59. This is an adjuvant which is licensed for use in Europe. It's been used there for 12 years or so. And I can tell you what it is. We can go to our trusty Google here.
Gotta love Google.
MF adjuvant is a submicron oil and water emulsion of a squalene, polyoxyethylene sorbitan monooleate, or tween 80.
Wow, tween 80.
You've used tween in the laboratory.
Of course,
so
I use it almost every day.
And sorbitan trioleate. It's a natural organic, squalene is a natural organic compound. And this was originally developed by Chiron, which is one of the original recombinant DNA companies years ago, and then acquired by Novartis. It's approved in Europe and found in several vaccines, influenza, and it's been licensed to other companies. So that's MF59. And this trial uses MF59, same virus as in the other clinical trial. This was done in the UK at the Leicester Royal Infirmary in Leicester, UK. And it was given to people with adjuvant, and they used different amounts, less vaccine. Let's see the numbers here. 7.5 micrograms, and 15 was the other. And that's in contrast to the other where they did 15 and 30.
And 30, right.
They assay by the same tests, HI and microneutralization, and they find that even the low dose here gives you good
7.5.
titers. 7.5 gives you good titers. They are also doing a non-adjuvant control, but that's not in this paper. And that's what I find funny about this.
They really should have included that.
Yeah, well, they should have waited, right?
Yeah.
Why do you think they didn't wait?
Well, it's just because it's swine flu.
That's right. They want to get it out
Yes.
first. And, it bothers me because I think you need to do the complete
Yeah, you really do.
study. But science is so competitive that you want to get your story out first.
Sometimes you just rush to publish and you skip the really important details. And a lot of things get lost in translation that way.
Yep. And you and I, of course, would never do this.
Oh, never.
Yeah, absolutely. We're smiling, of course. Anyway, so that's I'm interested to see the non-adjuvant to see if it's consistent with the other study.
Okay.
It's, of course, the 15 should be, if all is well. I mean, this is a different population. This is UK. And, they're different from the Australians.
Right.
Even though they have the same funny accents,
Right.
they are different. And the last paper is very interesting. Also New England Journal. All three New England Journal of Medicine. Just turning out to be the influenza journal of record. And this is from a number of groups. The CDC is involved here. And what they did was they wanted to know what kinds of pre-existing immunity is in humans against the new 2009 H1N1 virus. So they used a microneutralization assay to measure antibodies that were in stored samples.
I found this really interesting with the 1976 swine flu.
So they have people of different ages, 231 adults. Let's see. Let's read this. 55 children between the ages of 6 months and 9 years. They had 231 adults between the ages of 18 and 64 years, and 113 adults 60 years or older. These are all stored sera.
Right.
And this actually emphasizes that it's so important to have these banks of stored sera you can always go back to and answer questions. And they just took the serum and asked, do they neutralize the new 2001 strain? They do a neutralization assay. No HI here, just neutralization.
Sorry to interrupt you, Vince, but I got a curious question. Do they have sera from the 1918 pandemic? And could they ever compare the titers?
No, they don't.
No, that's just too long ago, and they probably didn't. I guess the preservation of that sort of things would never...
Yeah, that
technology wasn't good enough, I suppose.
I don't think we had freezers and refrigerators then.
No.
So it was very hard to preserve things. Like virus stocks were preserved by passing them in animals.
Right.
Until we got the ability to store them.
Or if you can find it in, let's say, there was this story that came out of somebody who died but got frozen in permafrost. And that person had the 1918 pandemic, which, as you know, killed so many billions of people. If you actually look...
Millions, not billions.
Oh,
right?
Sorry, yeah. Sorry about that. That would be bad.
I'm looking here at sera to see if we have anything. No,
it doesn't sound.
There isn't anything.
Yeah.
The lack of pre-1918 serum samples for analysis. It may be that there were some, but all that tells you is that, that those sera react with the 1918 virus that we recovered, because that's the only virus we have from back then.
Right.
It would be nice to have even older sera, but we don't. So in this study, what they found was that young kids didn't have any cross-reactive antibody, very low. Okay, 12 to 22% of the adults between 18 and 64 years had cross-reactive antibodies. Vaccination with seasonal strains generated very low antibodies, almost non-existent in children, and in 12 to 22% in 18 to 64 years of age, 5% or less in 60 years of older. But they found that vaccination with the 76 swine flu strain substantially boosted antibodies to H1N1 2009. So those
if you
people who got that
influenza
vaccine
back then, would you be...
In 1976?
Yeah.
Well, the strain at the time was an H3N2, so that wouldn't help. But you remember in 1976, there was a small outbreak of swine flu at Fort Dix. It never got outside the fort, but we made a vaccine and we immunized 40 million people.
Oh, okay.
And a lot of those people are still around. And in this study, those are the people who have the strongest cross-reactivity with the current strain.
Very interesting.
So, people are always very upset about that incident, and they say it was a wasted immunization. But now it turns out that these people 60 and over,
right,
whoever got the vaccine back then, are actually going to be protected against the current influenza.
So if you were sick in 1976, it's going to help you now.
Well, I'll tell you, in 1976, I was in Peter Palasi's lab, and when this whole thing broke,
By the way, I was born the next year.
In 77, so you didn't have swine flu. But this strain came to our department.
Oh, it did?
And we didn't know it. It was only years later that we were told that it was there. They were making a high-yielding recombinant for the vaccine strain. So I could have been infected with it, and maybe I'm protected. I should actually take my sera and check it. But anyway, I'm going to get the vaccine. So if you got the swine flu vaccine in 1976, it's sufficiently similar to the 2009 strain. In fact, I'm going to show you, Jason, just how similar it is. My colleague here at Columbia, Raul Rabadán, I said, Raul, does it make sense that the vaccine against 1976 swine flu would protect you against the 2009 strain? So he sent me a tree of the phylogenetic relationships, and I'll put some version of this online. But here's New Jersey 76, and here are the 2009 strains on the same branch.
Wow, that's pretty close.
So they're pretty close.
Huh.
And you can see all the other animal strains are quite different. Here's some other H1N1 strains. You can see they're very different. So it supports the notion that...
So I got another curious question. How come the antibody won't be cross-reactive to, let's say, seasonal flu? So I guess it's different enough that it won't see it.
Well, the H1 of the 2009 strain and the 1976 were swine H1s. So that's an H1 that had been evolving in swine. It's very different from the H1, the seasonal H1, which was evolving in humans. And these viruses actually evolve a lot slower in pigs than in humans. So the H1s were parallelly evolving in parallel in pigs and people.
Right.
Much faster in people than in pigs. And that's why the human strain, it's just too different. The seasonal human strain is too different, the H1. Does that make sense?
That makes sense.
Clear? All right, those are our stories. One more flu issue. The FDA on September 16th approved some of the H1N1 vaccines, the pandemic vaccines. And this is Sanofi Aventis, Novartis, CSL, which is the maker of the one that was in the study, and AstraZeneca, which is the MedImmune vaccine, FluMist. And Glaxo, I think, is the only one not yet approved. So these are approved, which means probably in a few weeks they'll start to be distributed, maybe as early as the first or second week in October. So I'm going to get it. Are you going to get it?
Well, here's a funny story. A couple of years ago, I did get a flu vaccine, but I got the flu a couple months later, and I was pretty sick. And I don't know if it was a reaction to the vaccine itself, because it was much longer or afterwards. I must have just got a different strain, rather than the seasonal flu. But this is when I was in grad school. So ever since then, I've actually shied away from
vaccines. What year was
vaccines.
that? 90s?
No, that was 2000, and must have been 2000 or 2001.
So it could be that you didn't have influenza, right?
It could have been.
Other viruses do influenza-like illnesses. Could be that year the vaccine didn't match well this strain. That happens sometimes, and you can get sick. So those are the two possibilities. But I can't tell you to get it, but
I'm probably going to go for it.
It's free here.
Yes.
You could just go get it. And I think it's worthwhile.
It's definitely worthwhile.
The more people that get it, the more will be protected. All right, so now the rest of the time we're going to spend on XMRV. But before we do, it's time for a word from the sponsors of this episode, Citrix, the makers of GoToMyPC. If you're a frequent traveler for business or pleasure, you know you still need to keep your business running when you're gone, and that means you need reliable access to your home or work computer. You can't take all the files with you. Wouldn't it be great if you could access it from wherever you are in the world? Well, the easy and secure way is GoToMyPC, brought to you by Citrix. And all you need is an internet connection to access your computer from anywhere. It's incredibly easy. All you do is go to their website, which I'll tell you in a moment. You load a small program, you get a login and a password, and then you're set to go. It takes less than 2 minutes. Then from any computer in the world, you go to their website, log into your account, and you will see the desktop of your computer on the screen. You can run any program on your computer in your office. You can open your email. You can access network resources. It's fabulous, secure, and incredibly simple. Right now, you can try Go to My PC free for 30 days. Just visit gotomypc.com/podcast. That's gotomypc.com/podcast for a free 30-day trial. We thank them for their support. Now, a couple of weeks ago, the news was about XMRV, which is a great name.
A couple of days ago, actually. Very recent.
And, yeah, it was a couple of days ago. And that's going to be the name of this episode. I love that XMRV. So what's this all about, Jason?
Well,
what does XMRV stand for?
It's a very fancy name. It's kind of confusing, but it stands for xenotropic murine leukemia-related virus. Basically, what that means is it's a mouse virus, murine leukemia-related virus. And the xenotropic is just a jargon for the cells that it can infect. So the virus actually looks like a mouse retrovirus, and but has gained the ability to jump species from mice to infect species other than mice. So it's xeno. It's basically gone away from the mouse.
If it just infected mice, what would it be called?
Ecotropic. So it would be an ecotropic murine leukemia-related virus. But
there's one other word, right?
Amphitrophic.
And that means?
It can infect any cell type.
Okay.
Mice, human, rats, rabbits, anything. So it basically looks like a mouse virus.
And this paper that we're talking about is called XMRV is present in malignant prostatic epithelium and is associated with prostate cancer, especially high-grade tumors. And it's from one of our former colleagues here, Ela Singh.
Yes, our lab actually worked with her a little bit on the virus, and we shared reagents and everything like that.
So maybe before we explain her paper, we should go to the original discovery of this virus. Right. Why don't you tell us how this was? This was done in the lab of Joe DeRisi and Don Ganem, right?
I think in California, correct?
California, yeah.
It's an interesting story, and it's basically why I'm doing my postdoc in Steve's lab.
Is that right?
Yep. Well, when I first came here 2 years ago, he came up to. I went up to him and I said, what projects do you have open? And he said, well, nobody's working on this one project, and it just came out. It's this new virus. It looks like a mouse virus. It's found in humans. It's probably nothing big. And I was like, sure, I'll work on that. And as time goes, things change. So I guess I'm in a lucky spot doing this research. But so the virus was discovered in 2006. It came out in PLOS Pathogens, and basically they worked with a prostate cancer group in Cleveland, and they wanted to see whether there were any sorts of viruses in specific type of prostate tumors. And they did this by doing a DNA microarray. So they took prostate samples, some that had tumors, some that didn't, and they isolated RNA, messenger RNA, and they queried all that messenger RNA for vironucleic acids. And this virus came up positive. But the most significant thing is that the virus was positive in these prostate tumors that had this mutation. And this mutation is in the RNase L gene.
What's that?
So RNase L it's predominantly identified as an antiviral gene, and it's part of the interferon-stimulated gene network, and everything goes back to interferons. But
you have good training, don't you?
Yeah. So in my PhD, we worked a lot on these sorts of things. So. Basically, when a virus infects a cell, that cell can actually recognize viruses. It can actually recognize different products of the virus. One of those products being the replication intermediates, such as RNA. Now, that cell that gets infected will probably die from the virus infection, but it's going to, at the same time, it's going to produce interferons, interferons are cytokines, which will signal to neighboring cells and get those cells to be resistant to virus infection. This interferon actually turns on the expression of RNase L. And it's important to point out that RNase L is just one gene out of 300 thousands of genes that are turned on in response to interferon. But the activity of RNase L is important because once the virus gets inside the cell, RNase L gets turned on, and it actually recognizes viral single-stranded RNA and degrades it. So the authors of this paper, they said, well, if the tumors already have this mutation, maybe these tumors are susceptible to virus infection. So they looked in those tumors.
So they had already known that the tumors had a change in RNA cell.
Right.
So they were wondering whether it would allow a virus to infect.
Exactly.
So what are the numbers? Does every prostate tumor have this RNA cell?
No, not at all, actually. It turns out this mutation is in about 13% of hereditary prostate cancer. So hereditary is a little bit different from sporadic prostate cancer in that you'll have certain other mutations that are associated with it in your family and everything like that. And RNA cell is one of the markers for hereditary prostate cancer.
So in this original study, what was the correlation between getting the virus from a prostate, or the sequence of the virus from the prostate tumor, and having the RNA cell mutation?
It's pretty high, actually. Mm-hmm. So if you look at one of the actually it says it in the abstract here. I believe 8 or 9 of the patients that were, it says in one of.
Here's 7 of 11 homozygous.
Right, that's it. Right.
1 of 8 heterozygous. And homozygous wild type. So
predominantly, XMRV was found.
40%, yeah.
Were found in prostate tumors that had this mutation.
So 40% of the tumors with the mutation, they say,
right.
Yeah.
Either homozygous or heterozygous.
So that doesn't prove it, but it strongly suggests
it's correlation.
Association.
Doesn't prove it.
Just like getting diarrhea when you get a flu shot.
Right.
Now, did they get actual virus from these tumors?
Well, they got viral nucleic acid. So that's where basically I came in. We collaborated with Don Ganem, and so Don Ganem gave Ela Singh, the author of the University of the Malignant Epithelial Prostate Cancer Story. She got the genome, and then she shared it with us, and we worked on it together for a short time. So no, they didn't get the virus themselves, the whole virus, but they can actually see in that initial story the cells that were infected and everything within those tumors.
So did they make hybridization probes for the virus and look in the cells of the tumors?
Yeah, they did it by a couple ways. They did it by fluorescent in situ hybridization, so they can actually see the nucleic acid of the virus in infected cells, and they did it by immunohistochemistry. So they looked at the proteins, the viral proteins that were present in the cell. And they basically took a slide from a pathological slide from prostate, and they could see it.
So what they say here is that they could detect nucleic acid and protein of this virus in 1% of stromal cells, predominantly fibroblasts and hematopoietic elements in regions adjacent to the carcinomas. Maybe you could explain what's a stromal cell.
Well, a stromal cell is a fancy word for a gamish of cells that, like, such as connective tissue cells, or cells that will basically, keep together the cohesiveness, the structure of a gland, your prostate gland, for example. So if you have a tumor, you're going to have a tumor growing from one site, and it'll grow out radially. But the cells around it that surround or encapsulate the tumor, those will be stromal cells. And they only found it significantly in only 1% of those stromal cells.
But not even in the tumor itself,
Not in the tumor. So very, very significant.
What does that mean?
Well, it could imply that the virus doesn't actually directly cause the oncogenic event or cancer. It just implies, doesn't prove anything, of course.
So it doesn't rule out its participation in tumor formation, right?
Doesn't rule it out.
Okay.
But, when this paper came out, a lot of people in the field, well, if it's only in 1% of the cells, maybe it's doing something else. Maybe it's generating, let's say, an immune reaction or an inflammatory state within that one cell that got infected, and then bringing in all the other aspects of the immune response to generate a proliferative state in cells, and then that maybe can cause the tumor.
Okay.
So just an idea.
Sure. Got it. All right. So based on that one publication, it looked pretty interesting. You started working on it.
I started working on it, and I actually don't do a lot of the work that they're doing. I'm more really interested in the virology of the virus, like how it replicates its life cycle in different cell types in tissue culture.
So you made a virus from the DNA, right?
Yes.
And I remember seeing you many times at lab meetings talking about that. that virus. And yes, you're interested in how it replicates in various cells. I think you were interested in the receptor also.
At one point, yes.
So so now we come to Ela Singh's paper. What does this add to the story?
Well, it adds a lot more, and it's pretty significant. Basically, she did kind of the same thing that the other paper did, in that they look at different prostate tumor samples, and they try to see within the prostate tumor samples where the virus is. They also try to correlate the incidence of XMRV infection with prostate cancer.
So they looked at 334 specimens. So I guess they say they're called resection. So I guess when you have your prostate surgically removed, they keep a piece, they store it, and here it's useful, right?
Right.
And they found protein in 23%, viral protein, DNA in 6%.
Right.
What does that mean?
Well, they basically looked at all different prostate cancer samples, and they either detected the virus either by PCR, looking to see whether the nucleic acid is there, or they did it by immunohistochemistry, by the protein, and they termed that as a positive hit. And that number, 23%, is from either one of those two tests, basically. And so it's 23% of prostate cancers and 6% that were controls, basically, also had XMRV, but much more in the prostate cancer.
So they also say the proteins were mainly in malignant epithelial cells.
Now, this is very different from the 2006 paper because, like I said, the 2006 paper had it only in stromal cells and very, very small fraction of that. Here, if you can see the figure, it's pretty clear. There are specific foci or groups of malignant epithelial cells that are really positive, that stain positive for viral proteins.
Mm-hmm.
So suggesting that the virus is actually in cancer cells as opposed to outside the cancer cell.
So here's some of these stainings that you're talking about, figure 2. Very nice.
Actually, I think it's the next figure. You'll see.
This one.
Yeah, it's a pretty striking figure.
Yeah.
You see a malignant epithelial section there, and the whole thing is pretty stains pretty brightly.
I also see some virions here. What's that all about?
Well, it's a new virus, so Ela Singh wanted to characterize the virus, and basically it looks like a retrovirus.
But these are not produced in the prostate tumors, though.
No.
These are in cell culture.
This is in cell culture. This is stuff that I do in the lab routinely, but she basically infected cells on a dish, basically, and allowed the virus to bud and release from the cell, and she took that and looked at it under electron microscope.
So has anyone seen these virus particles in someone's prostate tumor?
Not yet.
Is that not necessary to see particles?
Well, not necessary because, retroviruses, if you're going to see protein, chances are that the virus is already part of you. That's what retroviruses do.
Mm-hmm.
Just to quickly explain, retroviruses, they're RNA viruses, but when they get inside your cell, they kind of go backwards. They reverse transcribe their own RNA, producing DNA, and then integrate that in your own genome, basically, which becomes part of you. So that the staining that they see in that figure might not be virus particles per se. It just might be viral proteins that are expressed from cells that were infected at one point.
So does that mean some of the viral proteins are being expressed? All of them? We don't really know.
We don't really know.
So we don't know if there's an ongoing infection or if they're just a few proteins.
Right.
Maybe one of them is an oncogene of some kind being expressed.
It could be, though we don't really know. Classically, murine leukemia viruses don't have viral oncogenes. So in other words, there are many ways that retroviruses can cause cancer. Predominantly, the main way is that when a retrovirus integrates, becomes part of your host genome, it'll integrate in, let's say, a gene that is important for, let's say, cell proliferation. If it integrates near that, it'll turn on that gene, that proto-oncogene, for example, and then you'll get one step closer to cancer. That's the classic way of how these viruses do it. So.
Good. So I assume that in the cells in the tumors, the viral DNA is integrated.
Right.
They make mention in one of these, I think it was the first paper, that this DNA, this viral DNA, is not in the germline. Now, what does that mean?
Well, it's not in the germline. In other words, cells that are important for passing on your hereditary DNA, like spermatogenic genesis and everything like that. You won't. Supposedly, they don't find it in, let's say, cells in your testicles,
and ovaries,
testes, ovaries, that sort of thing.
So why is that important to know?
It's important to know from a general process that maybe this virus won't be transmitted genetically to your offspring. Although I don't know if that's generally the case, but that could be one important thing.
So I guess the implication is that these are viruses that we get by being infected, not through the germline. So that brings us to where this virus came from.
Well,
is it from getting it from mice, like hantaviruses, for example?
Well, at some point, we got it from a mouse. It jumps from mouse to human. How it got to other humans is topic of debate. There was a paper that came out earlier this year from that same group in Cleveland Clinic that showed that the viral nucleic acids is in semen, and so it's a possible mechanism.
Ah, interesting.
Yeah,
sexual transmission
could be possible.
So it somehow came from a mouse, but it could have gone to other species as well in between. We don't know, right? Are there other mouse retroviruses that infect us?
That's an interesting question. No, not per se, but the interesting thing is that I don't know if your audience knows this, but
you can assume they don't. So
retroviral DNA, retroviruses in general, actually take up a large part of our genome, and they're part of us, part of our existence, and everything like that. So I think something like maybe 10% of 5-10% of our genetic material is old retroviral junk. So we have a lot of retroviruses that are inactivated.
No, they're all dead, right?
They're all dead. They're all dead. They don't come out.
Or I should correct myself, because virus can't be dead because it's never alive, but they don't make infectious virus.
Right.
I have to be careful because I always...
Well, it's another topic for debate, I suppose.
I complain. Yeah, it is a debate. I have a very defined stand here, but when I talk about live virus vaccines or dead viruses, I have to correct myself because I'm making a mistake. But we understand they're not infectious, these old DNA copies of retroviruses in the genome. Are they related to murine viruses, or are they distinct?
They are related to murine. Like I said, it looks like it's a murine virus. It just has a different envelope that allows it to infect different cell types.
So maybe many, many years ago, these viruses infected us. And maybe even this... this virus is not in our germline, so it probably was more recent, right?
That's what people are thinking, yes.
Can we look in very old specimens to see how far back? Do we have prostate samples stored from more than 50, 60 years ago? 1918?
1918, for example. Yeah, we can get serum and prostate samples.
Well, that's how they got the virus from 1918. In part, there were formaldehyde-fixed, paraffin-embedded pathology specimens. So presumably...
As a matter of fact, I think your old mentor, Peter Palese, was involved in that project.
Yeah, he was involved with that. Because, he developed a technique to recover viruses from plasmids, influenza viruses, that is, not all viruses. So now this, the other thing about the Ela Singh paper, they say especially high-grade tumors are associated with this. Right. So what's the significance of that?
The significance is that they looked under immunohistochemistry, again, looking at the proteins in these slides, and they associated the presence of XMRV infection with a higher Gleason score. And a Gleason score is basically a characterization system that doctors use to characterize the extent of prostate cancer. So if you have a high Gleason score, you have a huge prostate cancer that's destroying the architecture of the prostate. And more XMRV positive samples were associated with higher Gleason scores.
So it could be that in the more serious tumors, this virus is preferentially involved.
Right.
So this is not a rare disease. This is 1 in 6 lifetime risk in the U.S. Globally, 3% of all men die of prostate cancer.
Yep.
So what has to be done next to get more proof that this virus causes prostate cancer?
Oh, a lot more needs to be done. A lot more needs to be done. More studies need to be done on. I would like to see like more of an epidemiological sort of thing. In other words, different populations, different centers, and if it's a mode of transmission such as sexually transmitted disease, then we're going to want to know what percentage in the population XMRV is in. Also, another thing is how pathogenic this virus is, how dangerous it is, because I've in the lab we're working with this virus, and when it came out, we assumed that it looked like a mouse virus, so it wasn't. I treated it like it was any other virus. You work with it very carefully, but this has been found actually, this virus, in normal tissue culture lines that prostate labs work with. So now a lot of people have to be very careful in handling it.
There is another interesting human tumor virus. It's called Merkel cell polyoma virus. You ever heard of that one?
I haven't actually.
So this is one of the human polyoma viruses that it seems to be oncogenic. It's associated with Merkel cell carcinoma, which is a skin cancer. Pat Moore and Yuan Cheng, who used to be here and who discovered Kaposi's sarcoma virus while they were here, have provided a lot of evidence that this virus might be involved in this carcinoma. And I did a blog post a few months ago about this, and Pat actually left quite an extensive comment.
Oh, wow, yeah.
And he said the most telling... so I had suggested that a good proof that this is a tumor virus, Merkel, is to put it in cells and show that it transforms them.
Right.
And he said, nah, that's not really good because a lot of things will transform cells, and it doesn't mean they're a tumor virus. He said you could put genes in and transform cells. He said the most telling evidence that a virus causes cancer is human cancer epidemiology itself.
I think it's very important to know the epidemiology of XMRV.
So that a lot of studies to see people with prostate cancer, do they have this or not? He also said in Merkel, the virus is monoclonally integrated into the cancer cells, and that I don't think has been looked at for XMRV yet, right?
Well, actually, it has.
Yeah.
People have looked to see whether where XMRV is integrated in the human genome, and you can predict that it's going to act like a mouse virus, and it integrates in sites that are open chromatin. And what I mean by open chromatin is that genes that are easily accessible, that are being turned on, being expressed, that are open, the virus will have access to and will integrate there.
Okay.
But it's not
monoclonal.
It's not monoclonal.
They have also shown that the virus is present in these cancers. The Merkel virus particles are actually present, which we don't have.
Don't have evidence for that yet.
But maybe we will have some. But those are the kinds of things. I think.
And it's interesting that you just mentioned that this this transformation experiment where you take a plate of cells and you hit it with a virus, and some viruses do that. They have these genes that will cause a tumor formation inside the plate, and you can actually measure that. So since I've been working with this virus, for so long, and I've never seen something like that. So I don't think the virus itself per se has a viral gene that might cause that.
So if you took Maloney, which is a mouse retrovirus related to this, and you infect cells, does it transform them?
No, it
does not. But there are other retroviruses like Rouse sarcoma.
Right.
Will make piles of transformed cells.
That's because those viruses have oncogenes that they've grabbed from previous infections and will generate a transformation. But XMRV doesn't have that.
It has to go. Maybe it has to go next to a gene in the cell and turn it on inappropriately to get transformation. Okay. What else? Anything else we should discuss about XMRV?
Not that I can think of. It's going to be a very interesting story to see how the more studies need to be done, basically.
So you're not doing any of this kind of epidemiology. You're doing molecular biology.
I'm doing a lot of molecular biology on the virus, and actually, I'm really interested in why XMRV loves prostate cells. So
it does love prostate
cells.
Oh,
it loves them really good. There's this one prostate cell line that we have that really replicates very well in it, and so I'm pretty excited. We got some very interesting data, and we actually submitted our paper a couple days ago.
Cool.
For it.
So that's a good sign that it grows well in prostate cells. It's consistent. It's another bit of evidence that it could be a prostate cancer virus, right? Well, we look forward to reading that when that comes out. That would be great. You want to see it in press. That's right. Do you think, what is your plan? Do you want to have your own lab someday?
Yeah, I would love to have my own
academic lab.
Academic lab. That's my goal.
Would you take XMRV and do work with it? Is that the plan, or you're not sure yet?
I'm not too sure yet, because I actually have other projects in the lab that are very exciting. So XMRV is just one aspect of retrovirology that I'm interested in. I mean, eventually my goal is to have my own lab where it's a general retrovirus lab looking at different aspects of virus-host gene interactions.
Okay. Very good. That's what I wanted to ask you. Now I remember. And now we can move on to email. And we have an email from Duncan. Now, Duncan is from South Africa, and he wrote a couple of months ago. He said there's an AIDS vaccine trial going on in South Africa. He said, why doesn't the press pick this up? So I said, well, there are dozens all over the world. And he said, I didn't know that. So he says, I sent him an article in IAVI, International AIDS Vaccine Initiative, I think it is. I have one right here on the desk. In the article, they say one challenge we have is to develop a vaccine. Another is to have a cure for AIDS. Isn't it misleading to talk about a cure rather than management, since HIV integrates within the host chromosome? I thought that cure would mean removing the integrated virus DNA, because even if you killed every HIV virus, the immune cells themselves would manufacture more HIV when they're activated.
Exactly.
So that cure would mean actually removing the integrated virus DNA. Isn't that impossible with our current knowledge of biology?
Well, and what he's referring to is called HIV latency. And basically, he's right. Once you get HIV and it infects your cells, like all retroviruses, they will integrate in part of your genome, and you basically have it for life. If you got rid of those cells, then you can cure it.
So that's what people are trying to figure out how to do.
That's what people are trying to do. There's actually a very interesting story of this one patient, somewhere in Europe, I forget where, that he had a very strong form of leukemia, but he also had HIV, and they basically gave him they killed off all his immune cells, and they gave him a bone marrow transplant from another donor that was CCR5 or one of the receptors that knocked out. So basically, there's a small population of humans that don't have the receptor for HIV, and a donor from that person.
That's right. So all his immune cells, he had a bone marrow transplant.
Right, and he was cured. He was
cured because there are no more cells.
There's no more cells. Yeah.
So even if you have all these latently infected lymphocytes, eventually they turn over.
They will turn over.
There's a long half-life, but they turn over, and there's no cells for the virus to infect. So eventually, you're cured. I've seen approaches where, the genomes are integrated into T cells. They activate those T cells to proliferate with IL interleukins of various kinds, and that eventually will kill those cells because the virus kills them. And then you hit with the antivirals at the same time to keep the virus down, and that's been explored as one other way.
It's effective, but again, all you need is just one cell to get by, sure. And it's almost impossible. So, in a sense, the person who emailed is correct. We don't have the current technology to do that. But although there are a lot of studies that are trying to address this very question.
Yeah, it's a great point, because even with long-term survival being very good, you still have genomes in you.
Exactly.
Like, the basketball player, Magic Johnson, he's healthy, but he still has genomes.
He might not have one virus particle in his body, but he still
DNA,
has the
DNA. By the way, I loved the last podcast on how a virus enters the cell. Have a great day. Duncan. Thank you, Duncan. Olivia writes, I just wanted to say that I love your podcast. I'm a first semester nursing student and had to take a basic microbiology course for a prereq, and I love it. I feel like I will eventually go back to study it more after nursing. But anyway, I just wanted to say that I really enjoy your podcast. With very basic bio knowledge, it's so informative and just interesting to me. Plus, a lot of the stuff you talk about obviously is important to the nursing field, too. So it's cool to see how they go together. Thanks. All right. I like to read praise letters, too.
Always makes us feel good.
Oh, yeah. Dell wrote, I recently discovered your wonderful virology podcast and have now listened to nearly every episode. Your TWiV podcast is entertaining and informative, an ideal combination, hopefully soon copied by other leading scientists. I'm a firm believer in the importance of cross-specialty sharing of ideas, something hard to do if the concepts are unnecessarily hidden by specialty jargon in esoteric journals or by sometimes semi-confused journalists of the popular press. You cut out the middleman, thank you. Although you never dumb the subject down too much, you do make a valiant effort to explain it to a layperson, and that is truly a gift for us mortals. Well, I'm mortal too, so...
I think it's really important to have a good communication system between the science community and the lay public. So, doing us a good service, Vince.
I enjoy it, but I wish some of my colleagues would take up the banner. I don't think it's going to happen, though. Next generation. So Dell is actually a physician from California. He goes on to say, if I have any good suggestions, one of them might be to take a few more breaks from H1N1 and adjuvants, which we got today, but keep us up to date on who is adding adjuvants and include more of the type of basic virus summaries with which you began this fascinating podcast series. Especially interesting to me are those viruses with human hosts, and if you aren't too afraid to add even more sex appeal to your podcast, please cover the sexually transmitted viruses, the kind that really make you sick. Yeah, actually, we have gone back to some basic virology in the recent months. And
have you done a podcast on HPV?
Not specifically, no. I'd like to do that. We have lots of suggestions, and that's one of them. I have to get somebody that... Well, actually, Saul Silverstein is a HPV expert, so we could get him to talk about it. Adjuvants. No adjuvants in the U.S. vaccine so far. Another suggestion might be to include a stronger clinical angle on some topics by inviting an infectious disease, OBGYN, or transplant specialist to your virtual table discussions of relevant topics. I think such a dialogue would stimulate discussion between both the scientists and the clinicians. For example, an in-depth discussion of viral structure and propagation of cytomegalo or herpes simplex or Epstein-Barr virus, potential causes of complication from infection or reactivation, including interesting related research ideas like superantigens, and ending in a discussion of the clinical effects to adults and children, including torch syndrome. What are some of the roadblocks to treating this virus that might be interesting for some young upstart to puzzle out? I do have plans to get more clinicians on the show, for sure. I have a couple lined up, so we will do that.
And you're at the perfect institution for it.
I do have lots of colleagues here, absolutely. For your consideration, I'd like to nominate a book as a weekly science book, Racing to the Beginning of the Road, The Search for the Origin of Cancer. This is by Robert A. Weinberg. So I haven't read it, but I shall. I shall take your suggestion, and perhaps it'll be a future pick. I read this book several years ago, and no doubt there are many more recent discoveries to shed light on the subject, but I found Racing to the Beginning of the Road to be a well-told, spellbinding story of the basic science underlying cancer, including the role of viruses and oncogenes. And then he has a P.S. My college roommate's mother, circa 78-9, developed Guillain-Barré syndrome and later died from the 1976 swine flu vaccine.
Interesting.
Apparently one of the approximately 500 fatalities you mentioned. I don't know all of the details, but I do recall that she was on a ventilator for some time, and the sad outcome of this experience caused me to avoid flu vaccines until I absolutely had to have them due to my work with direct patient care. I don't think there were 500 fatalities. There were 500 and some cases, of which some were fatal. I find it troubling that the real cause of GBS from the 1976 vaccine have never been found, but I do get the annual flu vaccines to protect my parents or other vulnerable people more than myself. I'm quite nervous about getting the rush order H1N1 vaccine, especially given the somewhat unknown adjuvant issue. Intuitively, it seems possible to me that somehow an adjuvant could aggravate any innate tendency to autoimmunity, which is what Guillain-Barré syndrome is. Thank you for shedding some light on this topic and at least mentioning the possibility of learning who is using adjuvant and who is not. Virology has sometimes grave real-life implications.
Well, this goes back to the whole debate on adjuvants, whether you should use them or not.
Dell, in the U.S., we're not going to have adjuvants, if that simplifies matters for you. But there's a very good discussion of adjuvants over at Effect Measure, which is a blog that we've linked to in the past. And you should go read that. It's very good. There's also a new page at CDC on Guillain-Barré syndrome, which we'll link to in the show notes. Vladimir wrote, Dear Vincent and great team, I'm writing to you from Florianopolis, a city in the south of Brazil. And first of all, let me thank you for your great program and podcasts. I'm a biologist who ended up getting involved in mathematical modeling of infectious diseases. As I work most of the year from home down here and keep in touch with colleagues and collaborators abroad, mainly through email on a daily basis, so I miss this sort of personal interdisciplinary chat that run in coffee breaks in scientific departments and that are so stimulating and enlightening. Your podcast brings the fresh air of that sort of environment each week, so thanks for that too. I have a few questions regarding influenza antivirals that I'm curious mainly due to two aspects, public health policy and an understanding of how the immune system reacts to a viral infection. I would much appreciate your input on this. First, if someone takes the antivirals, let's say 6 hours after the beginning of influenza symptoms, has the immune system time to fully trigger the process of antigen recognition? I.e., is this person vaccinated against that strain of influenza?
Now, I'm going to take a guess. I think yes.
It doesn't completely eliminate viral replication.
So the immune system would see virus.
Right.
I mean, there'll just be a lot less.
Yeah, you might have a less robust response. Maybe you wouldn't have good memory. I think you would have some, because my reading of this is that the antivirals don't completely inhibit.
No.
They just bring it down enough so that you reduce shedding and you reduce symptoms.
Right, and you have to start using those antivirals pretty early, as I recall, right?
No later than 48 hours after the first symptoms.
After that, it's kind of pointless.
No, it's not worth taking because the infection is going to go anyway.
Yeah.
2, if the person is feeling well after the intake of antiviral, something that seems to be the case even after an hour, and keeps the treatment going, is it still possible that he/she can transmit the virus to others? If so, for how long would you estimate? So this is a tough one because, in fact, this hasn't been done with H1N1 very much. But with the other strains, it doesn't. It reduces transmission for a day or two, but it doesn't eliminate it.
Right.
I think we have to emphasize that you're still shedding. You're shedding less, and that may reduce the efficiency of transmission.
I've got a curious question for you. At what point do you shed the most during the actual infection?
So you can shed a day before symptoms.
Okay. And
then you can shed up to 5 days. So there's a peak at three days or so.
Right.
But again, that's an average. Everybody is slightly different or can be different.
See, what I think is significant for this question is that I think you can shed even before the onset of symptoms.
Right.
So you might not be taking any antivirals, and you still might be shedding, or transmitting virus.
Yes, that's right. So where you start taking the antivirals is crucial, of course.
Yeah.
But I wouldn't have a false sense of security that if you're taking antivirals, you can walk around and interact.
Still got to wash your hands and cover your mouth.
I think you have to be careful. Yeah. 3, if symptoms persist even after a timely started treatment with antivirals, can we say there is a non-neglectable chance that the virus has developed resistance? Hence, more drastic contingency measures should be applied to this person, like monitored isolation in biosafety installations?
That's a very good question. There's always definitely a chance for mutagenesis with influenza, and specifically recombination with different viral genes. What's your take on it, Vince?
Well, there is resistance spreading to all of these influenza viruses to Tamiflu. So if the patient doesn't respond, it's either not flu or it's a resistant strain, in which case you're not going to have much of an effect on the course. I think you should take sensible precautions, but I don't think you have to be isolated. Influenza is not one of those diseases that requires isolation or is really impacted very much by isolation. It might be the case that based on this kind of information, some guidelines could be refined to adjust the period of quarantine for patients under antiviral treatment to avoid transmission to others and at the same time prevent the so feared development of virus resistance. Thanks again for your time and great podcasts. Well, again, quarantine is not really going to happen because there's so much contact with other people that it really is ineffective. Couple of PS, just in case there are some time left. No problem if not. Well, we have a few minutes near the last email. PS1, by the way, in case it might be of interest, a colleague of mine has suggested to me a website called FluWeb Influenza Historical Resources Database that is hosted by the School of Population Health, University of Melbourne, and that is built on the nice idea of compiling epidemiological information of past pandemics. It struck me as interesting as I am right now supervising one student on digitizing and analyzing the death certificates of the 1918s and surrounding years from cemeteries and municipal archives of our hometown.
That's interesting.
I began to realize that this sort of research is a great and inexpensive way of introducing new students to the epidemiology field at the same time of rescuing a source of highly interesting and frequently forgotten and vanished information. That's cool.
That's really cool.
It could turn up some interesting stuff, right? And then it's available to everyone. P.S. 2. I translated from Portuguese this information for Dick, who likes to follow cases of rabies. In the Secretariat of Health of the Brazilian state of São Paulo, which is crossed by the Tropic of Capricorn line, they found that out of the 675 cases of animal rabies found in 4 years, roughly 50% were present in bats, 40% in bovines, 10% in horses. There were also 5 cases in pigs and 1 in a dog with a bat virus, and he puts a link for that, which we'll put in the show notes. Great. Thanks very much, Vladimir, and thanks everyone else for your emails. That'll bring us to Picks of the Week before we wrap up, and I asked Jason if he could bring us a pick because I'm sure he's got something interesting. What do you got, Jason?
Well, it's kind of interesting. When I was in the lab the other day, and another friend of mine, postdoc in the lab, he showed me this website for this artist. His name is Luke Jerram, and he's in the UK, and he does really cool stuff. He actually, I'm always interested in merging of art and science, and he makes glass sculptures of different viruses and bacteria and everything. So it's called glass microbiology. It's his work, his pieces are actually they look really beautiful, and you can actually see clearly through different viruses and see what like the capsid of HIV would look like, and all these things on a microscopic level you can actually see. And these huge sculptures. Definitely check out the website.
It's got a movie of him doing the glass sculpturing with a blowtorch.
Yeah, it seems cool. It's really hard to do, but
it's got HIV, smallpox, swine flu, of course. Yeah, that's beautiful, and the pictures are gorgeous. We'll post that. That's wonderful. Thanks for picking that up.
Yeah, no problem.
That is exactly the kind of pick we love here. Luke Jerram. Well, my pick is actually the website recommended by Vladimir, the FluWeb Influenza Historical Resources Database. We'll put the link to that. And as he said, they're collecting information from all over the world related to flu. But the one thing on here that's very good is there is a book. It's a monograph published by the British Ministry of Health in 1920 regarding the 1918 pandemic. 600 pages and contains over 100 tables with data never before analyzed about the pandemic. So the whole book is here online, and you can look at it.
Oh, wow, cool.
So that's a great resource. Plus, the whole database is growing daily. So check that out, the FluWeb Influenza Historical Database, which is thanks to Vladimir. And that will do it for another TWiV. Jason, I really appreciate you joining us.
Oh, no problem.
I know you have great experiments waiting for you out there, so an hour is a big deal.
Actually, I have to go back to the lab right now and do some more XMRV experiments.
Well, believe it or not, I'm going to do a plaque assay now. He doesn't believe me, but I am.
No, I've seen you in the lab, Vince.
I still love doing experiments. We appreciate your discussion of virology. You fit in pretty well, and so we would like to have you back sometime.
Oh, yeah, no
talk
problem.
about your other work, talk about Hendra and Nipah
virus.
That would be great.
Absolutely.
Thanks a lot. You don't have a website, do you? A personal website?
No, I don't.
Okay, because Dick de Pommier, of course, has a web.
Unfortunately, I can't shamelessly plug myself, I guess.
That's okay. When you have a lab, then you'll plug yourself.
Yeah, that's true.
But TWiV, of course, has a website, twiv.tv, where you can find show notes and links to all the stories we talk about. And do send your questions and comments to twiv@twiv.tv. You can send an email. You could send an MP3 file. Or you can call up Skype at TWiV Podcast and leave a voicemail. And we always like hearing your comments and questions. And we're a bit behind. I'm sorry about that, but we're trying to catch up. But we read just about every one because they're all excellent. And you've been listening to This Week in Virology, the podcast all about viruses. Thanks for joining us. We'll be back next week. Another TWiV is viral.